course, just like when the next Q&A starts, let's plan that the Q&A in the chat is over before the Q&A for the next session starts. Yeah, if that's that's a good idea. And let's just start with our talk. So the first talk we have today is by Sankalp and Satvik, both are undergrads at the when the next Q&A starts, let's plan that the Q&A in the chat is over for the Q&A for the next session starts. Yeah, if that that's a good idea. And let's just why can I the talk. So the first talk we have today is by Sankalp and Satvik, both are undergrads at the next. Is it just me or can you hear what I just said?
Yeah, there seemed to have been a glitch.
Wow.
So the YouTube link is also on. So I thought it was a YouTube link that was playing in the background, but it seems there's some glitch.
The first talk we have today is by Sankalp and Satvik. Oh. I mean, this will continue even when the speakers talk, right? Okay, stopped, I think. So the first talk is by Sankalp and Satvik, both are undergrads at the Biotech Department at IIT Madras. The talk is titled Picasso, Cellular Automaton for Spatial Organization. So yeah, over to you, Sankalp and Satvik.
Okay, I'll just share my screen.
Hi everyone. Can you see our slides? Okay.
Yes.
Hi everyone, good afternoon. I'm Sankalpa and along with Satvik, we're presenting our work Picasso, a cellular automaton for spatial organization. It was originally inspired by a talk we had the opportunity to attend earlier this year at the India EMBO meeting, and we had the opportunity to work on it with Dr. Karthik Raman in his course, Computational Systems Biology. So going into the talk. Natural microbial communities have been shown to exhibit spatial organization, and it's been seen in soil aggregates in the plant rhizosphere, for example, and we've illustrated this in the kelp microbiome as well as the human skin microbiome here, and this has also been observed in the human gut microbiome and oral microbiome. And all of this evidence points to the fact that it may have some functional implication. However, the study of the spatial organization is limited to the inherent complexity of the system, which necessitates a synthetic microbial community setting to study this patterning. And these synthetic communities are also useful because when you have large genetic circuits, which are modular, and these large genetic circuits have a particular function, one major limiting factor is the crosstalk between different components of this genetic circuit. And in this case as well, spatial organization can be used as a tunable parameter to separate different modules of these genetic circuits into different cells and study them. So we set out to model spatial patterning in microbial communities, and we did this in the form of a cellular automaton. And the cell division was modeled as a stochastic process in this case. And in our models, microbes can communicate either via coherent sensing molecules or through the use of essential metabolites. And I'll talk more about this in detail. And our models overall provide a theoretical basis to understand the self-organization of cells in a microbial community. So coming to the first model, this is well understood and characterized. It's where you have two auxotrophic strains. So in this case, you have a blue strain and you have an orange strain. And the blue strain does not produce an essential metabolite, say the orange metabolite. And as you can see on the right, these strains are obligate mutuals, which means that in the monocultures, in the absence of the other strain or in the absence of the other metabolite, it does not grow. And they grow only in the co-culture, which is how communication is engineered here. And the model that we propose that is built upon an AHL device library that was characterized by Dr. Polizzi's group is based on codon sensing. So as you see here, represented for two strains, you have the first strain that produces the AHL molecule, which is 3OC8HSL, represented by blue pentagons. And when this is taken up by the second strain, it causes the expression of a particular gene that increases the fitness of the second strain. And we'll explain how this happens. But you see that this is also true in the case of strain 2, where strain 2 produces these pink circles, that is PCHSL, and this similarly increases the fitness of strain 1. And this mode of orthogonal communication can be extended to multiple strains, and a representative figure here indicates it for 6 strains. So in this case, the blue pentagons produced by strain 1 affects an gene expression in strain 6 that leads to increased fitness of strain 6, and so on and so forth until strain 2 similarly can increase the fitness of strain 1 when strain 1 senses the AHL produced by strain 2. So coming to how we went about choosing these particular genes that can actually increase the fitness, we used a flux balance based strategy for this. So what you see here are representative results using the genome scale metabolic model, IAF1260 for E. coli. So what we did was, from the genome scale metabolic model, we performed a single gene deletion and shortlisted, and we calculated the growth rate, and the growth ratio is essentially the growth rate divided by the wild type growth rate, and we filtered for those that had a ratio between 10 and 90%. So the genetic background of the strains we consider don't have these genes. So it has a single gene deletion for one of these genes, and upon codon sensing, the wild type levels of this gene are restored, which leads to essentially rescue of this gene, so the fitness increases.
So our models are available as a MATLAB package on GitHub, and we have also included templates that can be used to generate whatever configurations the user wishes to. So this is the workflow within the package. So first the user inputs a metabolic model, a genome scale metabolic model, after which the package generates possible gene lists that the user can choose from on which gene that is to be deleted. The user then selects the genes. The user must then input the generation time of the organism or the strain being considered, based on which the package computes the fitnesses both of the single gene deletion mutants as well as of the wild type. The Polizzi lab, along with their large library of AHL and device pairs, also has published a MATLAB package that can be used to select optimal AHL device pairs for whatever purpose is being considered. And we propose that that package can be used to select the codon sensing device, which can then be input into our package. Based on the AHL device pair chosen, the package computes the threshold concentration of codon sensing molecules that are required to cause the expression of a gene. And the model may then be run either for predefined configurations that are present in the package, such as, for example, microbes that have been seeded in parallel lines or in concentric circles around each other. And as mentioned, there are also templates that allow the user to input any seeding configuration of choice and see how it grows. So the blue boxes in the flowchart represent steps that are carried out by the package itself, whereas the brown boxes represent steps that require user input. The package can be run starting at 2 points. In case you're working with a new organism, that is anything other than E. coli, or you wish to use a genome scale metabolic model other than IAF1260, you would have to input a new model and start the run from here. In case you wish to proceed with E. coli IAF1260 itself, you can automatically start the process from this step. We wanted to validate the diffusion scheme that were used in our model, which is a forward time central space scheme that is detailed here. So we decided to try and replicate results from a popular paper that in synthetic biology that studies morphogenesis, which is Basu et al. So Basu et al. Built these plasmids that encode what is essentially a bandpass filter that leads to the expression of green fluorescent protein at AHL concentrations between 10 power minus 2 and 2 into 10 power minus 1 micromoles, which they modeled and then validated in experiments. So what this sort of a bandpass filter essentially creates is, if you have an agar plate and you have a center colony shown in cyan here that is constantly secreting AHL molecules, you end up with a ring of green fluorescence protein that corresponds to the region in which the AHL concentration falls into this band. And when they measured this, they found that this pattern takes 36 hours to equilibrate. And you end up with a ring that's between 5 millimeters and 18 millimeters from the center of the center colony. We used many of the same parameters for the bandpass filter that they used, and we used a lot of parameters for the diffusivity in agar of the metabolite and so on that are taken from literature. And we were rather surprised. We found that our model almost exactly recapitulates what they see in the same time frame, that is over a period of 36 hours. So rather than 5 to 18 millimeters, our model gives you an output of 3 to 18 millimeters for the dimensions of the ring, which is because the model is coarse grained, and each of these little blocks that you see corresponds to a distance of 3 millimeters. We further wanted to see what would happen if we tried these various configurations that Basu et al. Had attempted as well. Basically, there is a lawn of cells encoding the bandpass filter on the plate, and they place center colonies in various different sizes and configurations on the plate. And we found that these can also be qualitatively replicated by our model. So this is what a run being seeded randomly across the plate. In these runs, what you a useful thing to remember is that each cell block is 3 millimeters long. Each iteration or each step in the model takes a time of 10 minutes. Uh, there are around 10 power 8 cells within each cell block. And the total grid size that we've used in all our simulations is 30 by 30 centimeters, but that can, of course, be modified as you wish. We then decided to do a grid search to sort of explore the parameter space. The 3 parameters that we choose to study are the 3 that are most amenable to experimental variation. So these are the rate of secretion of quorum sensing molecules, which can be varied by using promoters of different strengths to drive the expression of the enzyme that synthesizes the AHL molecule. The fitness gain, which can be modulated by choosing different genes based on the FBA results, and the threshold, which can be changed by choosing different AHL device pairs based on the library available from the Polizzi lab. In each of these cases, there are 2 strains that exist on the plate. There's a red strain and there's a blue strain. And in each of these cases, we are varying the parameter for the red strain along an experimentally amenable range of values along all 3 axes. And we're keeping the parameter for the blue strain constant at a median value. And each point in this, and we're comparing the populations of each strain at the end of the run. So there are 125 different parameter sets being considered, and for each parameter set, we ran it for 50 steps, which corresponds to 500 minutes, and these are averaged over 100 different runs for each parameter set. So each circle, or rather each sphere here, represents the color of the sphere represents which strain shows a greater population on average at that parameter set, and the size of the sphere is proportional to the difference between the populations of the two strains. For example, a large red circle would indicate that at this parameter set, you get more cell blocks corresponding to the red strain. And there is a fairly large difference between the populations of the red strain and the blue strain. A small blue dot, like here, would indicate that you get more blue cells than red cells, but the difference is not all that significant between the two populations. The most obvious trend that we noticed from this is that as you vary the rate of secretion, uh, as you go from this side of the box towards this side of the box, you see a gradual shift from where you see a gradual accumulation of blue in the box, which indicates that as you increase the rate of secretion of AHL molecules from the red strain, you see an advantage in fitness being derived by the blue strain. And we wanted to investigate these effects in greater detail, which is why we did some more fine-grained analyses, which Sankalpa will talk about.
So, as Karthik mentioned earlier, we ran each parameter set for one iteration, that is 500 minutes, and did this 100 times to take the mean value. So this time for each parameter set, we let it run for 1000 minutes and we saw what the processes were like. So as Karthik explained, what we have here is the red strain. Strain 1 was varied. Its rate of secretion of QS molecules was varied, whereas that for strain 2 was kept constant at the median value. And what we found was when strain 1 secreted more QS molecules, which is found in the later part of this graph, strain 2 enjoyed an advantage, which makes sense because they're coupled. And this was found also in the earlier part of the graph when strain 2 secreted more colon sensing molecules than strain 1, which led to an advantage enjoyed by strain 1. And these results were also seen in the three-strain model, where the strains are orthogonally coupled. So here strain 1 is coupled to strain 3, strain 3 is coupled to strain 2, and strain 2 is coupled to strain 1. So when the rate of secretion of strain 1 was higher, that is at the second end of this graph, we see that the green strain enjoys an advantage. We then looked at the variation in threshold concentration, and we saw that early on, when there is a very high threshold needed to affect gene expression, there is for the blue strain compared to the red strain, which is much lower here, the red strain enjoyed an advantage, but the process seemed to equilibrate, and this was observed even in the case of the three-strain model. And so this further supports our conclusion that it is the variation in the rate of secretion that has greatest sensitivity in this model and is probably most useful to vary for experiments. And when we also looked at the gain in fitness, we found that the processes seem to show no real trend, and this is probably due to the stochastic nature of our models. So what we wanted to look at next was whether all these values that we saw was actually a result of model behavior and not of model breakdown. So we did this by coming up with a preferential growth assay. So here we have two strains. The red strain and the blue strain are seeded in two parallel lines. And what we quantify is the number of red cells that proliferate towards the blue cells and the number of blue cells that proliferate towards the red cells and those that proliferate away from each other. And we looked at this for all of our parameter sets. So first, for the rate of secretion, in these graphs, the colored blocks represent the cells that proliferate towards, whereas the black boxes represent the cells that proliferate away. And we see that overall, across parameter sets, there are more cells that proliferate towards rather than away from the strain to which it's coupled to, both in the case of strain 1 and strain 2. And this trend is seen when we also vary the threshold concentration, where on top you see that more red cells proliferate towards blue cells, more blue cells proliferate towards red cells rather than away. And it was also seen in the case of when we varied the fitness gain, where more red cells proliferated towards the blue cells rather than away. And this is seen in strain 2 as well. But this gave us confidence that the cells that we saw were not in fact due to model breakdown, and the cells and the two strains did remain coupled throughout these experiments.
So the preferential growth assay is useful to see whether or not your model is broken down. It's also useful in a sense to quantify the strength of coupling that you see in your model. And we wanted to compare the two models that were introduced at the beginning of this talk, which are the quorum sensing mediated model and the metabolite mediated model, which is why we performed preferential growth assays using two strains in both of these models. And you see that in case of both models, both strains more preferentially grow towards each other than away, which indicates that there is coupling present in both the QSMM and the MMM. However, it is very clear from these plots that there is much stronger coupling in case of the metabolite mediated model than in the case of the quorum sensing mediated model. And intuitively, this makes sense because in case of the quorum sensing mediated model, the strains you're working with, in the absence of their mutualistic counterpart, are essentially strains that have had a gene that helps in growth deleted, which reduces the growth rate from the wild type value to some suboptimal value. However, there is still a positive growth rate here. In case of the metabolite mediated model, the strains that we're using are auxotropes of an essential metabolite, which causes a much greater disadvantage as far as growth is concerned compared to a deletion of some non-synthetic lethal gene. So the fitness of each strain here in the absence of its mutualistic counterpart is practically zero, which would explain why there is much stronger coupling in case of the metabolite mediated model. However, stronger coupling does not necessarily mean that it is the best model, which we'll come to in a little bit. So to conclude, uh, To conclude, we first validated the diffusion scheme that we're using in our model by replicating results from the classic paper, Basu et al. We then packaged all our code into a MATLAB package that's available on GitHub and can be used to study spatial organization in microbial communities. And it's extendable to different species and different quorum sensing frameworks. We then analyzed the two different methods that we're considering to engineer communication between strains, and we found that the metabolite exchange mediated model allows for tighter coupling. However, it is also true that the quorum sensing mediated model is a lot easier to use when you're trying to extend communication to more cell types. This is because you don't have to deal with numerous interdependent auxotropes here. You can instead simply deal with fairly modular AHL device pairs that have been designed and validated already. We proposed genetic circuits to validate our models, and we proposed strategies based on flux balance analysis to select genes that can be used in these circuits. The design of our circuits that you previously saw are based on previous work from the Polizzi lab. And we found that when there is the greatest sensitivity here is for the rate of secretion of quorum sensing molecules. And if you wish to alter the behavior of the system, or you wish to modulate the behavior of your system along some range of parameters, or you wish to see a range of behaviors, the best parameter to vary would be the rate of secretion of quorum sensing molecules, which can be achieved by using a library of promoters for the enzymes that synthesize the quorum sensing molecules. So to summarize, we first proposed genetic circuits to implement a new form of communication that we're proposing, which is the quorum sensing mediated model. We then built a model that can simulate the behavior of a system that has communication using quorum sensing in this manner. And we ran the system for two strains as well as three strains, and we analyzed the behavior in each of these cases. We're very grateful to Professor Karthik Raman. This project started off, as Sankalpa mentioned, under his computational systems biology course, and it and yeah, he really helped us along the way. We'd also like to thank Gayatri Samamurthy. She was very helpful with regards to some of the ways in which we could plot our simulation results. We also had a lot of fun working on our project, and we're also really grateful to everyone who was there in the course with us for all their support. These are some of our key references, and thank you. We open the floor to any questions or comments.
Thank you, Sankalp and Satvik, for a nice talk. Do we have any questions? We don't. So okay, I have a question. So in the diffusion scheme, so like, can you go to the
Yeah.
So like just on the grid, like how many? So if yeah, you can just like stay with any grid that is there. Oh yeah, take this grid. So like take the blue box. Like how many boxes closer to this box can say AHL or any metabolite be like diffused to?
It could theoretically keep diffusing through the entire grid, although how far it actually gets depends on how long you're running your model. So each step in the model is for 10 minutes, right? And the parameters are based on physical data from experiments, and they've been calibrated for the time steps and the length steps that we're using.
Okay, so like the diffusion parameters are from experimental papers.
They're
Yeah.
all physically relevant values. Yeah, they're not assumptions.
Okay, this is true both for AHL and metabolites, or
Yes, it's true for both AHL and metabolites.
but for metabolites, I think there were two papers recently that show that the diffusion scales are pretty small. So like uh, maybe yeah.
Um, I'm not sure about that. So this is based on experiments where they constructed polyacrylamide gels and they injected metabolites and they simply measured its diffusion across the thing where no cells are present. There are of course going to be slight variations depending on the
Oh, okay,
density of the
okay,
agar itself and
okay,
on the
okay.
cell density and stuff, but I'm not sure about.
Yeah, that's okay. Thanks. Yeah. Do you have any other questions? Oh, we have a. Okay, yeah. There is a question which says why the median constant.
Um, so here, right, like in all of these, basically.
Yeah.
So what we're doing here is that you could vary the parameters for both strains simultaneously. That is doable. It just will take a lot more time. So what we did was, out of the two strains that we're studying, we just kept the value for the parameters for one of the strains constant, and we varied it for the other. I mean,
Yeah.
you could do it for both, of course. Like, yeah, I mean, that would give you more in-depth results, I guess.
Yeah, but this. Yeah, I guess this makes sense too. Uh, okay, so I think that was it. Thanks guys for nice talk. For nice talk.
I'll stop sharing.
Yeah. So yeah, so the next talk, we'll move on to the next talk. It is by Nilesh Ananda Subramanian, who is a postdoc fellow at IBSE. And his talk is going to be on systems biology of diabetes Hellman's interaction study. So disease interaction, a systems approach to disease interactions. And yeah, take it away, Nilesh.
Is my screen visible?
Yeah.
Okay. Good afternoon, everyone. I'm Nilesh. I'm a postbachelor fellow working with Professor Manikandan. So today I'll give a talk on the project that I'm working on, which is systems biology of disease interaction, a diabetes Hellman's as a case study. So disease comorbidity is, well, if you have to define comorbidity, it's defined as a disease occurring on top of a primary disease. So primary disease of interest, like example, it could be something like diabetes and TB, or cancer and sepsis, or HIV and AIDS, or HIV and hepatitis,
Nilesh. Oh, Nilesh, you are muted and you can't see your screen.
Sorry, yeah, just yeah, sorry. Is it visible
Yeah,
now?
it is visible now. Yeah.
Yeah, sure. Okay. Um, so example of disease comorbidities is something like diabetes and Hellman's, or HIV and TB, or cancer and sepsis, or something very much recent like COVID and diabetes. So here, why it is important to study is, I mean, especially the biochemical and the immunological variables involved in it and their interaction, because two diseases may function in a very distinct way, but when it occurs separately, but then when it occurs together, they might behave in a different way. That was in fact important to understand the multiple diseases that are occurring together. So in this image, as you can see, it's like a generalized representation of this protein-protein interaction network, and the sequential network changes, depict the physical protein-protein interaction rewiring during disease progression. So like a combination of different perturbations, like a disease risk variant, or genetic or environmental perturbations, or sometimes even a treatment for some particular case, can alter the protein abundance, and therefore in turn lead to rewire of the network topology. And especially if that is if the disease modulus are bridged by say a central node, or different many other nodes, or that are like common between the two networks, then we can then we can assume that they are considered they're considered to be important in the comorbidity. um, so basically, these kind of studies are enabled by systematically generated data sets. Like example, we have like about let's say you have like 4 groups of data. Like you need to have like a control and the data set for just one disease, then the two disease, then both of them occurring together. Then such analysis can be carried out. Um, yeah. So just to make sure that we are all on the same page, I would like to spend some time in defining few immunological terminologies before I go into the work, since I would be using a lot of these terms in the rest of the talk. So starting with inflammation, which is a complex biological response of the body tissue to any harmful stimuli like pathogens, dead cells, or irritants. And whenever we have inflammation taking place in any part of the body, we'll have immune cells that are associated with it. And these immune cells produce cytokines, which are basically proteins that are helpful for intercellular communications. Now, if you look at the immune cells itself, they can be classified depending on the type of immune system, which is if it's like an innate, if the immune response is innate, then cells like eosinophils, basophils, neutrophils, or macrophages are involved. If it's like an adaptive immune response, we have cells like B cells, T cells, and in T cells itself, we have T cytotoxic and or T helper cells. And in this particular talk, we'll be primarily focusing on the T helper cells, like we have Th1 and Th17, which are primarily involved in the pro-inflammatory immune response, whereas the Th2 cells are majorly involved in the anti-inflammatory response. Now, we all know that diseases involve biomarkers to characterize them. So similarly for helminth infection, one of the biomarkers is IgG, which is actually an antibody, immunoglobulin type G. Then in case of diabetes, we have like HbA1c, then RBG. RBG is like a random glucose measurement. So these are like considered to be the biomarkers for diabetes. Now, so these are the examples of certain cytokines of these types of TH cells that I mentioned earlier. So we have like interleukin 12, interferon gamma, TNF, tumor necrosis factor alpha are like the examples of this TH, that is cytokines that are produced by this TH1 cells. Similarly, cytokines that are produced by TH2 cells are like IL4, IL6, and 13, and so on. Similarly, for IL-17, cytokines produced by IL-17 cells are like IL-17A, 22, and so on. So if you look at Helminth infection as such, then it's mainly caused by strongylosis staphylococcus, which is a parasite that is transmitted from soil, and we all know that parasites, they require a host for survival and reproduction. And such infections usually elicit immune response from the body. In this case, it is a Th2 type of response mainly. Now, if you look at diabetes, various genetic factors and environmental factors are involved in diabetes, and but then a major aspect with respect to this type of diabetes is something known as insulin resistance. We all know cells require glucose for energy, so insulin is required for glucose uptake. In insulin resistant conditions, cells don't respond to insulin well and can't use glucose. Therefore, over a period of time, blood glucose levels increase, uh, and eventually they lead to a hyperglycemic state, causing diabetes. Usually, pro-inflammatory cytokines are highly active in diabetic conditions. So these diseases have been well studied separately. Many pathways and regulatory networks and pathology of the disease have been studied extensively. But then the studies that look into the interface or study the comorbidity of both the diseases, of both the diseases is something that has been studied like it's very low. And so out of these few studies, we can see that human and animal studies have demonstrated that Helminth infections are associated with decreased prevalence of type 2 diabetes mellitus. So such observations, people have made such observations, and so this is like the still there is some sort of uncertainty that is involved in the interface of these two diseases, and so this is like the motivation for our work: study the interface of Helminth and diabetes and study their interaction. So through our work, we try to shed some light on the interface region of both the diseases. In this talk, we look at the overview of the data set. Overview of this, the data set that was used to understand the disease interaction. Then we'll also look at the interaction analysis that was performed to identify the DDI that is the disease interaction markers. Then we'll also see how these disease interaction markers are connected to one another using patient networks. We'll also see on a differential correlation study to see the effect of Helminth's treatment on the rewiring of the disease relations. So first, let's look at the data set. So this is like the general overview of the data set. So here we can see that two sets of data, that is for patients without diabetes and with diabetes. So here T2DM minus suggests that individuals, they don't have diabetes. So under each of these two sets, we have three groups, mainly control, infected pre-treatment and infected post-treatment with respect to Helminth's disease. We have about 60 individuals individuals in each of these groups. We have about 50 variables, which are like the biochemical parameters that has been broadly classified under such variable categories, like that is depending on the background of the variables. For example, if you have certain measurements like adiponectin or adipsin, leptin, or resistin, and so on, they all broadly come under the classification of adipocytokines, that is the cytokines that are produced by adipose tissue. So, so depending on the background of the different variables, we just classified them, which will be later useful for us while interpreting the results. Now let's look at the interaction analysis. So to understand, so the main aim here is to understand the interaction between the two diseases, diabetes and Hellman's, and also to identify important markers that are involved in disease interactions. So basically different linear regression models are built to conduct such interaction analysis. And the samples that we are particularly looking at are basically the control and the infected pre-treatment samples uh, from the DM minus group, DM minus set of population. Then we have the control and infected pre-treatment groups from the DM plus group individuals. Here, the whole interaction analysis itself can be split into two segments. One is like the main effects or differential expression analysis, as it is popularly called in bioinformatics. Then we have, which is basically to study the effect of one of the independent variables on the dependent variables. Whereas interaction effect is like, so interaction occurs when the effect of one independent variable on the dependent variable changes depending on the level of another independent variable. So the independent variables might interact with each other. So it's like the interaction effects indicate that a third independent third variable influence the relationship between the independent and the dependent variable. Thereby, the output itself is influenced by both the independent variables. So there's just like a general flow chart over here that has been done. So here, basically, it's like a two-pass filter. Like initially, we perform the main effect analysis and filter out those variables that are said to have main effect, and then perform interaction effect analysis on the filtered out variables to obtain a set of variables that is said to be influenced by both Helminth and diabetes. So below here is an example for one of the variables that was found in the interaction effect, which is interferon gamma or IFNG. So here we have like a box plot and an interaction plot. So from the box plot, we can see that for for different combinations of information regarding the diabetes and Helminth, like for example, you can see like no Helminth and diabetes minus, Helminth and diabetes minus, no Helminth and diabetes plus, and Helminth and diabetes plus. For various such combinations, we can see how there is this huge variations in the distribution, and we can also one can also observe that in the last final combination, that is having a Helminth and a diabetes, we can see there's a huge positive shift with respect to interferon gamma, so suggesting that there is interaction effect happening. Another way to look at or visualize the interaction is through interaction plot, which is more commonly used. So typically non-parallel lines indicate interaction effect, and we can clearly see that here. A way to represent the results from the interaction analysis is through a ternary plot, which is a ternary plot with the three vertices being like Helminth, diabetes, and Helminth diabetes, Helminth diabetes, that is an interaction term. So the relative proportion of Helminth diabetes interaction and Helminth and diabetes variance is shown for the variables in the data. So this plot captures the relative proportion of variances that is explained by these three terms on the variables. So if a point, say if a point is at a vertex of a triangle, then the relative proportion of variance due to the labeled factor is 100. So the dots are the variables, as you can see, and I have labeled a few of them like IL-17A, IFNG, IL-13, IgG, leptin, and HbA1c. So the black dots or black points over here, they are the ones that show only main effect, whereas the points that have like a green green color around them show interaction effect also. And as I said earlier, the points that are closer to the vertex are influenced heavily by that particular label. And we can see that biomarker like IgG is close to the Helminth vertex, and biomarker HbA1c is close to the diabetes vertex. So this is like one such observation that we could see. And also the ones in the center are said to have like effect on them by these three terms. So interaction plot that explains how these variables are being influenced by both the terms. And I have explained this with respect to interferon gamma. So I'll just quickly go through some of these plots that I've shown here. For example, in case of IL-17A, or be it interferon gamma or IL-13, we see that the non-parallel lines basically indicate that there is an interaction that exists between them.
Hello,
Similarly, if you
Nilesh, can you move the chat window from over the presentation? I think it is.
Sorry, just a second.
Nilesh, what happened?
Just a second, sir. Is it visible now?
Yeah, can you go into presentation?
Yeah.
Yeah, I think it looks good now. Yeah.
Yeah. Okay. So yeah, as I was saying, for IL, these are just few examples that I'm showing here, like for IL-17A, interferon gamma, IL-13, or even if you consider like IgG, though the interaction is like very small, it still comes as a significant interaction. And if you look at something like leptin or HbA1c, it's like almost like parallel lines, which suggest that there is no interaction, and that's what we also see here, just the black dots. So next we will look at the Bayesian network that helps to connect the diseases interaction markers. So typically Bayesian networks are used to construct networks of interaction between biochemical parameters or molecular factors, and to observe the connections among these factors. So especially, and in a case like ours, where we have more than one disease that affects a person or an individual, then it's of interest to find how these molecular networks get affected under different disease background, that is in a comorbid condition. So this is just a basic skeleton of the Bayesian network that was generated with the 4 groups that I mentioned earlier. So let me just highlight some of the DDI, diseases interaction marker nodes here in this network, so that it's easier to visualize. So I have just highlighted some of the DDI markers here. So I have highlighted the ones that have like a relative proportion of variance higher than 40%, that is from the ternary plot, and these are the names of those nodes that I have mentioned here, just for people to see. And from here we can see that all these DDI markers, they tend to appear together, and they seem like they have like dense connections with each other, indicating like a stronger role in the interface of the 2 diseases. Also, we included 2 terms here, that is this D and H, which mean D for diabetes and H for Hellman's. So this is something that suggests that Hellman's is it's something that captures the essence or the information of the samples that have Hellman's infection and that don't have Hellman's infection. Similarly, D is like a node that captures the information of the samples that have diabetes and that don't have diabetes. One interesting thing to notice here is the biomarkers like IgG is it's like has a direct connection with the H node, that is for the Hellman's node, and we also have like the biomarker RBG, random blood glucose, is also directly connected to the D node. So this is something interesting, and it kind of signifies that these terms, that is the D and H terms, have like a direct effect on the biomarkers, as we can see here. So this particular analysis is just for us to see how these DDI markers as such are like connected in general. Next, we'll look at the differential correlation study. So consider a variable pair, say variable A and variable B, and in a control population, they said to let's say these 2 variables have high correlation that exists among them. So now let's measure the same variable A and variable B from an infected set of population. Because of the infection as such, they could have completely lost their correlation that was existing between them. So let's say we treat these patients and after a period of like say 6 months after the treatment, we again measure these variables, that is variable A and variable B. So there are, so either it could have, so there's a possibility that it could have reverted back to its original state that was there in the control set of population, and such kind of a pair is known as a resolved differential correlated pair. Similarly, another thing can also happen, which is, let's say you have variable C and variable D, and they're said to be highly correlated in the control population. In the infected state, they could have completely lost their correlation, and even after post-treatment, they could still remain the same way. That is, they could have, they did not revert back to its to their original configuration. So this is an example of an unresolved DC pair. So what I mean by here is like we specifically search for pairs of variables whose correlation has increased or decreased in strength in the infected group compared to the control group. So this is what we define as a significant differentially correlated pair. Now we further investigated whether the differential correlation of these variable pairs is resolved or unresolved in the treatment group. So this is just like a workflow where we use different methodologies like bootstrap and equivalence testing to identify the significant and the resolved and the unresolved DC pairs.
Nilesh,
Hello.
Nilesh, it's almost like 20 minutes. Can you?
Yeah.
I'm sorry to interrupt. Yeah.
Sorry, I'll complete it fast. Yeah. So this is an example of a DC pair where so this is a correlation plot of one such DC pair that was reported as unresolved in the DM plus conditions between the TH17 cytokine, that is IL-17A, and TH1 cytokine IL-2. We can see that there is we can see the change in the direction of correlation, that is like a positive correlation in the control to the negative correlation infected and the treatment groups. Positive correlation of these pro-inflammatory cytokines are expected in the control group, which consists only of only the diabetic individuals. So upon helminth infection, this correlation is disrupted, maybe through the activation of anti-inflammatory cytokines, and that's why we see such observations. And we don't see such changes in the non-diabetic individuals. So we can see that the differential correlation of these pro-inflammatory cytokines are not resolved even after helminth treatment. So this is like a so in case of DM plus, this is like an example of an unresolved edge or an unresolved DC pair. So this is a summary of the number of significant resolved and unresolved DC pairs that we find in the DM minus population and in the DM plus population. So I'll just conclude quickly. So the diseases interaction markers like IL-17A, 13, 4, IL-2, TNF-alpha, and so on were identified using the interaction analysis. Ternary plot and interaction plots were used to visualize the effect of helminth diabetes interaction on the variables that were identified as BDI markers. Then using Bayesian networks, dense interconnections among the DDI markers were visualized, suggesting that they play like a very stronger role in the interface that exists between the two diseases. And through the differential correlation study, significant resolved and unresolved pairs of variables were identified that suggests that the rewiring of these variable relations is something like an effect because of the helminth treatment. So finally, I'd like to acknowledge Dr. Manikandan Narayanan, sir, for his constant support and guidance. I'd also like to thank Philji, Dr. Philji, who is also a part of this work. I'll also thank Dr. Subhash and lab members from the NIRT Chennai for providing us with the data set. I'll also thank Words group members for their constant input in the group meetings and IBSE. Thank you.
Thanks a lot, Nilesh, and I'm really sorry for the interruptions. We have like a minute for taking any questions. Yeah, so one of the questions is if this comorbidity of helminth and type 2 diabetes is there, will it affect other, like, will it cause other related disease like hyperglycemia and hypoglycemia, or does it only affect type 2 diabetes?
So, I mean, hyperglycemia is like it's already like a part of type 2 diabetes itself, right? So, uh, so
Okay.
I'm really, yeah.
Yeah. So the other question is, how does one assign and verify a given if a given interaction exists?
Sorry, hello.
Oh yeah, the question is, how does one assign and verify if a given interaction exists?
Okay. So we could see from the ternary plot that those, for example, IgG and HbA1c, which were like the biomarkers for helminth infection and the diabetes, so they could be found in their respective vertices, right? So which itself like a validation for us saying that okay, these markers are identified near the vertices. So well, we still have to uh, probably take a deeper look and see how these interactions play out specifically for each and every variable.
All right, and just like a quick question from another attendee, that what did the y-axis represent in the interaction plot?
The y-axis is basically the variable of interest, like if it's
Okay.
like an interferon gamma um, or IL-13.
Okay. Yeah. Thanks a lot, Nilesh. We'll move on to our next speaker, who is Dinesh Kumar. He's a MS student, and his talk is going to be on computational analysis of ocular microbiome in bacterial keratitis infected eye. Over to you, Dinesh.
Hello everyone. I hope everyone can see my screen.
Yeah. It is visible. Yeah.
Okay, let me just change to pointer. Okay. So, hi everyone. So my talk is going to be about computational analysis of ocular microbiome in bacterial keratosis infected eye. So let us right away go into. Before going into ocular microbiome, let us see what is microbiome. Uh, so basically, microorganisms are present everywhere in skin, gut, soil, the places you see, places you don't see, like deep sea. So everywhere. So one of the most interesting things about this microbiome is that they exist as communities. So when you say communities, it means there is a lot of interactions occurring between each and every microorganism. So so why do they have to live in communities? Why do the microbes have to live in communities? There are lot of advantages in living in communities, such as like they can have a division of labor, that every individual does not have to do every job. The, the job is basically shared between all the microbes. So this also improves the metabolic capabilities, the advantages of other microbes also gets now added up to the community. So like when when you have four different advantages, and when these four different microbes form a community, then the community will have all these four advantages. So these are all certain aspects, certain features of a microbe being in a community. Okay, so okay, how can we study these microbial communities? So we say that these microbial communities play a lot of role in a lot of diseases. Uh, there are lots of recent researchers in gut microbiome, soil microbiome, a lot of things like that. So how do we study this? So step one is we need a metabolic network. We need a network. So what is a metabolic network? Again, metabolic network is just, so as I have shown in this figure, is just an overview of the metabolism, every reaction pathways happening inside a microbiome. So there are tools. There are these metabolic networks can be constructed from the genome of the organism. So the present recent technologies have enabled us to uh, like obtain the complete genomes of a lot of microorganisms. So from these genomes, we can use these tools such as K-base, Kawami, Model Seed. These tools help us to construct these metabolic networks. We call these metabolic networks as genome scale metabolic models because, as I said, they are constructed from the genome of the microorganism. So it is just the in silico representation. So when you want to do a computational analysis on a microbiome, you need like the genome scale GSM of all the individual microbes present in the microbiome. So after that, once we have these genome scale metabolic models, and then what we do, and then we use those models to analyze the microbial communities. There are like basically two ways of analyzing. One is the constraint based modeling, constraint based analysis, where we have like a we have this metabolic network, and we have an objective function, which is basically the biomass, and then we have a um, linear equation. We have a stoichiometric matrix. Uh, and then we put in certain constraints in order to based on the when we put in certain constraints based on how we want um how we want the objective function to work. So this is basically a flux balance analysis. This is a constraint based modeling. Then the other one is the network based modeling. Network based modeling is just when you have a genome scale metabolic network. What you do is so you just you like we can what you can do is you can try to give in some seed metabolites and try to see uh, mm, what are the metabolic capabilities of these models. So, so in this presentation, I'm mostly going to concentrate on network-based modeling. So, network-based modeling. Okay. So we had this algorithm, this is a MetQuest, which was developed by Dr. Ati Ravikrishnan in Computation Systems Biology Lab in IIT Madras. So, so it is available. It is a Python-based package, and it is available in GitHub and Raman Lab. So, okay, let me tell you what this MetQuest does. It can basically do two things. Like it can construct bipartite graphs with these metabolic networks. So bipartite graph is basically the graph of the whole metabolism in so where you have like a graph made up of reactions and metabolites. Uh, the metabolites leading to the reactions. So this is basically the bipartite graph. So MetQuest, this MetQuest algorithm also does another function where it can walk through when you give a certain set of seed metabolites. This it can walk through the bipartite graph and see, and it can like so for example, in this figure, like let's say that S1 and S2 are the two seed metabolites being given as input. So when it is given as input, it basically walks through this all these reactions R1, R2, R3, because all these metabolites required for these reactions are present. So but when it comes to the reaction R4, this metabolite M4, which is not present, hence this reaction cannot be executed. So what happens is this reaction. So the reactions that are executed are considered as visited reactions. They are reactions that which could not be executed. It comes under list called stuck reactions. So this MetQuest algorithm basically gives us an output, a list of list of visited reactions and the list of stuck reactions, and it can also give the as you said, the metabolic capability of a microbe, like the what all metabolites can be produced and secreted by this.
அந்த full dump download பண்ணிட்டு recompile பண்ணி
So it can also give us an output, the set of metabolites that can be produced and secreted out by the microbe. So 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 we have this Metquist algorithm. Can we use this Metquist algorithm to study the microbial interactions? Yes, we can. And how? We devised mm, index called as MSI, Metabolic Support Index, which is basically a fraction of stuck reactions relieved by the presence of another organism. Okay, so let me explain this concept a little more. So, for example, let us consider this microbe A, which has like some 50 reactions, in which these 1, 2, 3, 4, 5, 6 reactions are stuck. So, let us also consider another microbe B. When present individually, it might have like some 60 reactions, in which these 4 reactions are stuck. But when these two microbes are put together, what happens is the number of stuck reactions is reduced. You can see that in A it has been reduced from 6 to 2, while in a B the number of stuck reactions is reduced from 4 to 3. So considering this concept, we this index, metabolic support index, is being devised. So this is based on the so as I said, this is the fraction of uh, stuck reactions which is relieved in an organism in the presence of another organism. So this is basically like the property of one microbe to us to like interact with another microbe. So how are these reactions, how are these stuck reactions being relieved? So when this microbe A gives some metabolite to this microbe B, which could not be produced, which is not available to this microbe B earlier, but now it is available because this A can give that particular metabolite to B. So that's how this stuck reaction, the reaction that is stuck earlier, is now relieved. So this metabolic support index gives us an insight of whether a given two microbe can interact or not. What might be the interaction between, so what metabolite is being shared. So all these things can be uh, obtained from this metabolic support index concept. So applying this method to ocular microbiome data. So we claim that, so we can so let me tell the what we can see. Like we can, I'm telling that we can see the network structure of ocular microbiome in terms of MSI, and we can also predict the key players in the microbial community, in this ocular microbial community. The major, like if we take the major pathogen responsible for this bacterial characteristics. So with this MSI, I'm telling that we believe that we can predict the key players in a microbial community. We can also see the structure of a microbial community. So let us see if we can do that. So after microbiome data, so so this is so let me tell you what data, what after microbiome data is. So this data is being it basically consists of the it's basically relative abundance data. So it consists of the list of all the microbes that is present in infected individuals. Data collected from 29 bacterial characteristics infected individuals, and the healthy microbiome data collected from 36 healthy individuals. So it comprises of all the microbes present in infected individuals and healthy individuals, and its relative abundance. So this data is provided by Dr. Shivaji from LV Prasad Eye Institute, Hyderabad. So So basically, what is bacterial keratosis? It is an bacterial infection in the cornea of eye. It causes eye redness, eye pain, blood vision, and it also causes corneal ulceration. So the main thing here is it causes dysbiosis of eye. Dysbiosis is the disruption in the microbiome of eye. So let us not just explore more into let us not just go into the literatures of bacterial keratosis. We'll just have this. Instead, what we'll do is we'll just obtain results and inferences from this experiment, and then we'll try to compare the inferences obtained from this MSA experiment, and try to compare it with the data already available, so that we'll see the capability of this MSA experiment. So here's the workflow. What we do is so we have a set of microbe is a set of microbes having abundance above a particular cutoff, like a 0.1 percentage or 1%, 0.1% cutoff. So all these microbes will be the genome information of all these microbes will be considered, and metabolic models will be built. So with these metabolic models, we'll try to determine the dependence of one microbe on the other. That is the metabolic support index. So we'll calculate pairwise metabolic support index of all these microbes, and with this we'll construct a network. With this we'll construct a network. So we'll now see how the network will look like. So the result of the legend, I will explain the legend also with the network. So this is basically how an MSA pairwise network will look like. Okay, this is a octal microbiome network of BKCR patients. So BKCR, so there are like two ways of collecting samples from an eye, from an infected eye. One is corneal scrapings, which is BKCR, and the other is corneal swabs, which will be represented as BKSW. So we can see it is always happy. It will be always nice to see a network, but when so there are like 30 microbes present here, and hence uh, this network might be a bit complex to look at. So we'll have this heat map, which is which is which is the which is the same network, which is represented as a heat map. So we'll try to obtain some inferences from this heat map. So we can see this cluster here. So what does this cluster tells us? So how much how much these microbes benefit by the presence of the microbes present in the bottom. So for example, so in the presence of this microbe, Pseudomonas aeruginosa, this microbe, Streptococcus thermophilus, will be benefited uh, by a MSA factor of 0.166. So 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 that's how we can look at this heat map. So we see that. So whenever the table is dark vertically, it means that the below the bottom supports all the microbes in the left hand side. So you can see that the 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 column corresponding to Klebsiella acetoca is a little bit dark when compared to other columns. So this says that this microbe Klebsiella acetoca is supports all the microbes present in this bacterial keratosis community. So So let's see. So the microbes being benefited from these Klebsiella oxytosa and these Pseudomonas species. So Streptococcus pneumoniae, Streptococcus aurelis, Streptococcus cristaceus. So we have a set of different microbes being benefited from these two major microbes, and we also see a dark row, which means that this bacteria, Pyomonas Pyomonas methylalpha-alphatogenes is getting support from it can get support from all these microbes irrespectively. So we can say that this microbe has the ability to get metabolites from all these microbes. So this is about bacterial catalysis, corneal scrap, corneal scrapping. So now let's move on to the other sample, which is bacterial keratosis sample obtained from corneal swabs. So this is the network for it. So likewise, we'll look into the thing. We see a big cluster here, which corresponds to microbes like Klebsiella. So all these belongs to Klebsiella. So we have Klebsiella michigansis, Klebsiella oxytoca, another strain of Klebsiella oxytoca, and we have Klebsiella pneumoniae, and we also have this Lecretia species. So there is another thing, which is the corresponding to Pseudomonas aeruginosa. So you can see that in the previous thing we had only one Klebsiella oxytoca and lots of Pseudomonas species, but in this sample we have like a lot of Klebsiella species, while we have only one like one or two Pseudomonas aeruginosa. Like we have only two Pseudomonas species. So let's see the microbes being benefited here. So these are the microbes being benefited here, which are almost the same as those we saw in the previous thing. Uh, we have this Capnocytophagia sputogenica, which was not present there, and we have this Haemophilus influenzae. So okay, so and these as we saw this horizontal dark columns there too. Here also these microbes, which are the most benefited, like which can benefit from any microbes present here, like all the microbes present in this community. So this being the results of bacterial characteristics, let's see the microbiome of the healthy.
Sorry to interrupt, Dinesh, but we are 20 minutes into the presentation. Can you please?
Yeah. So it's done, almost done.
Okay. Oh, nice. Okay.
So in this ocular microbiome of healthy, we can see that we have E. coli here. We have a Pseudomonas psychotolerance here, and we have Streptomyces canis here, which supports a lot of organisms. Okay, so we have seen everything. Let's try to infer what we saw. I can say that more than one Pseudomonas, as you said before, there were more than one Pseudomonas species in bacterial corneal BKCR, and only one Klebsiella species was present. Whereas in BKSW, it was the opposite. So we had only one. We had like more than one Klebsiella and only one Pseudomonas species. So on the whole, this Klebsiella and Pseudomonas collectively supports all these microbes. So like what we can assume is that it is okay to assume that Klebsiella and Pseudomonas are key players in bacterial catalysis microbiome. The majorly these two microbes, because they were present in both the samples, the BKCR and BKSW. So we can also consider this: the organisms supported by these Klebsiella and Pseudomonas were also pathogenic. So inferences from the healthy microbiome. So we can see that the E. coli, Streptococcus canis, Pseudomonas cyclotrons were the major supporters of the healthy eye. And there was also a decrease in when we see this particular bacteria, Streptococcus pneumoniae, which had a relative abundance of 11.29 percentage, but in healthy microbiome it was only 0.34 percentage. The same for Pseudomonas suginosa, aeruginosa, which had 13.95 abundance percentage in bacterial characteristics microbiome, while less than 0.1 in healthy. So, what caused this decrease in abundance? So, we have to we have to look deeper into these microbiome data in order to find the answer for this. So now let's see what the literature says. From literature, it has been seen that the bacteria, the microbes, Pseudomonas aeruginosa, uh, so all these four microbes were most common pathogens of bacterial characteristics, which like which goes with what we had predicted. So that's how we
Hey,
come at a
Dinesh, hurry up,
conclusion,
Dinesh.
sir.
Hurry up, hurry up, hurry up.
Yes, sir, done, sir. So that's how we conclude that MSI experiment can be useful in studying the microbial community. So acknowledgement, I thank my guide Dr. Karthik Raman for support and ideas. I am grateful for Sanjay Balakrishnan. I would like to acknowledge Sanjay Balakrishnan for the work together in this project, and would also like to thank Aarti Ravikrishnan and Marcia Ibrahim for their help during this process.
Thank you, Dinesh, for the nice talk. We just have a minute, and there is one question. I'll just.
Sure.
So the question is, what happens if an organism in the sample doesn't have a metabolic model?
So no, like we have to construct metabolic models. The main thing is here is that we have to the organism should have a complete genome. So we should have the genome information of a model. So in case if we do not have the genome information of a particular organism, we can, the best possible way is to blast the 16S rRNA and try to get very closer, try to find closer microbes which can be used in its place.
Okay. Yeah, that I think that answers that.
Yes.
Okay. Thanks a lot, Dinesh, again. And we'll move on to our next speaker. Our next speaker is Pavan Kumar, who is an MTech student. His talk is going to be on local Genie, a novel thresholding algorithm for developing context-specific models from omics data. Over to you, Pavan.
Yeah, thank you, Shreyans. Is the screen visible?
Oh yeah, it is visible. Yeah, it is visible, Pavan.
Yeah, thank you. Thank you. I was just. So hello and good afternoon, one and all. I am Pavan, doing my MTech in Biotech department. I'm here to give the talk on local Genie, a novel thresholding algorithm for developing context-specific models from omics data. And this work is guided by Dr. Nero P. Bhatt from Biotech department. So this is the general outline of the talk which I'm going to give today, giving the introduction to the metabolic networks and following through why we need context-specific metabolic networks, and where thresholding methods helps in developing these context-specific models. Actually, threshold methoding is a pre-posing step, and what are the drawbacks in the existing thresholding methods, and we try to develop this new thresholding methods called local thresholding, and compared with the existing thresholding method to show in the results, and followed by the conclusion and future work we have to do. To begin with introduction, computational models have been built from biological and computational knowledge. to just test some hypothesis and to develop the deeper understanding and generate new insights. One such computational model is called a genome scale metabolic network. Yeah, my previous talk, the previous talk by Dinesh was also based on this. I will add some more points what genome scale metabolic networks is. So it's a reconstruction that consists of metabolic information, means all the reaction information of organism, and compiles it as a mathematical model. Some of the examples of human genome scale metabolic networks are this Recon 1, Recon 2, Recon series goes on, and Edinburgh human metabolic network. These networks, genome scale metabolic networks, basically comes with these. There might be some additional information, but basically it comes with these three information, which is here R1, R2, R3 just reactions, and for this particular reaction to occur, gene 1 or gene 2 can be present, which are basically isoenzymes. And similarly, for R2 occur, gene 1 and gene 4 has to be present, which means they are subunits of a single enzymes. So both genes has to be expressed for reaction R2 to occur. This is one information we'll have in the genome scale metabolic network. Similarly, we have stoichiometric matrix, which here ABCD are metabolites, and these are the reactions. So if negative signs means they are substrate and positive means they are products in that particular reaction R1. Similarly, it comes with constraints where we say that flux through this particular constraint must be within this range. So if we have our lower bound to be negative means what it tells, it is a irreversible reaction, and if it is positive means it is a reversible reactions. So all generally the genome scale metabolic networks comes with these infos. So where do these genome scale metabolics used is just to predict the cellular behavior by changing the constraints under different biological conditions, and to design drug targets and metabolic engineering strategies, like optimize a particular reaction to develop a particular product, and develop the and engineer the corresponding reactions which are developed. But do all the reactions defined in this genome scale metabolic networks are present in every human tissue? Means genome scale metabolic network, as we shown before, this Recon and Edinburgh have all the reactions of the which occurs in humans. But if you take a particular context, which is, for example, if you take brain, it will have a particular set of reactions, which means a subset of reactions which are defined in this genome scale metabolic networks. Not all will be present. So this is where we need to develop context specific models, which will be a subset of this genome scale models. So for example, if we have brain, only the reactions which are present, which are to be there in the brain, are alone taken from this genome scale model. Obtaining this context specific models. So we have our genome scale models in the hand, and there are many algorithms developed to filter out the reactions which are context irrelevant, and to get this context model finally. So algorithms basically requires has these requirements, which are constraints, as we defined before, this flux constraint, which we can give if you have exometabolics data, or even if you have domain knowledge, we can go on giving the constraint. And almost all algorithms require some reactions to be defined as active reactions or context relevant reactions. Some algorithms ask for scores, some ask for give core reactions, and so active reactions, the way of asking this active reactions depends on the algorithm, but almost all algorithms ask for this active reactions. If we know about the context, or if we have some defined knowledge, it's easy to define this active reactions. But if we don't, we need help from this omics data, from where we have to define this active reactions. These active reactions, to be obtained from the omics data, we have to give a threshold. Like we have genes expression, we'll use the GPR rules to choose some active reactions. So how using this omics data we'll get these active reactions? Basically, the existing thresholding methods, what it does is give a single threshold, and whatever the genes cross that particular threshold will be considered to be active, and below will be inactive. And this goes on basically on assumption that all genes have the same expression pattern but this is not the case. For example, if there is this housekeeping reactions and enzymes, which even though their genes expression value is low, they have to be present in all the context for basic cellular maintenance. And there are some stable mRNAs which may have this low expression gene expression, but they must also be there in the context. So to give a small intro to existing thresholding methods, this is global threshold. Global threshold is nothing but the thing which I defined before, like we'll give a single threshold to the omics data. So the genes which cross the threshold will be considered to be the active, and which are below them will be considered to be inactive. So what if we have more info, like single context gene knowledge? If we have, means we have to rely on global threshold only. But if we have a set of context expressions values, means for we consider example where we have a 2D array or a matrix where the rows are genes and the columns are the context. So for each row, the genes expression values across the context, we have this values. So if can we able to use this info, like for a particular context, in a particular context, a particular gene, how it is expressed in that context and how it is expressed in the remaining context. Using that info, there are some other thresholding methods developed in this. The first one is the local. So here, this box plot shown here is this gene one's expression across the context. Means the 2D matrix which I defined before, that 2D, in that 2D matrix, a single row is this box plot. So in this box plot, what local thresholding goes on is just simply take the mean or a median, some middle Weshel value, and give the threshold, apply this threshold to this, and whatever genes crosses this threshold are considered to be active, else inactive. Similarly for all other genes also. But the problem with this local thresholding method is, consider a gene where its expression values are low, like 0 and all. So there are chances that there are chances that some context will include this gene to be active. And similarly for higher expression genes also, where a particular gene has its expression level, has an expression level as a higher value in all the context. In that case, what what it does a mistake is, it's filter out this reactions even though they're highly expressed, because it comes below the mean value. A certain context expression values as comes below this mean value, they are filtered out. So then came local T2, where along with the local specific threshold, we have to give two more threshold. One is upper and lower, which says that if gene's expression value crosses this upper threshold, thereafter it will be included in that context. Here the mean will not be its threshold. Similarly, if genes similarly if gene's expression value goes below this lower threshold, it will be not included in the context. Even though it's mean, it may cross the mean value, but if it didn't cross this lower threshold, it will not be included in the included to be active reaction set. Then came this stand-up method, which is a recently published one, where what I do is check the patterns of expression patterns of this genes and cluster them together and give a cluster specific threshold. So all the genes present in a particular cluster will have the same threshold, and that threshold they just calculated by getting the mean and standard deviation of the data. We found that there's a room for development in these cases, like to infer more details about this relative thing. How we can infer more by taking a particular context gene expressions and other context gene expressions. So local Gini, which I'm going to define later on, is based on this Gini coefficient. This Gini coefficient, let me explain what it is first. Gini coefficient or Gini index is a measure of income or wealth. Gini coefficient was basically developed to measure the inequality in the income measurement of a particular country. So how it goes on is, this is the basic formula. Like there will be a comparison between each and every person's income, how much there is here how much there is an inequality between them, and finally we'll get an index which lies between 0 and 1. If the Gini coefficient is 0, means we have equal distribution of income in all the peoples in that particular country, and if it is 1, means only one guy has the whole wealth in that particular country. This is the nice expression which I have of Gini coefficient in the Wikipedia. Um, Here this line of inequality, after arranging the income of persons in ascending order, so as we add on people, the cumulative income is also must also increase in this alignments. There is equal income, and we have our Gini coefficient to be 0 there. And defining this Gini coefficient in this terms means it's nothing but a divided by a plus b, area of this a divided by area of this a plus b. And Gini coefficient based threshold was developed similar something like this. So this was something, as I explained before, like if Gini coefficient is 0, all contexts have same expressions, and if Gini coefficient is 1, this particular context will have have its Gini expression, and all other will remain 0 there. So we have our Gini coefficient values in the hand, which lies between 0 and 1. Those values are converted into percentile values, and these values were applied to this to this genes. So we have a 100 context, means we for a particular gene we have 100 expression levels for this 100 context. So we'll apply the percentile value and get the threshold, which is the gene specific threshold, which is local Gini threshold. Here local Gini also faces the same problem faced by this local threshold, where there are chances of excluding highly expressed reactions and including low expressed reactions. So similar idea was taken from local T2 by saying that we are going to give 2 more thresholds, like upper and higher. Similar, so idea goes on basically like local T2. This is the complete flowchart of what we have done. We have taken 2 data sets. One is for cancer cell line data set and tissue data sets, which these are the contexts basically, and process the data set. As we defined before, we had this GPR rules, Boolean rules, and RAR. GPR rules are used to just to make a gene expression mapping to the reaction. So we are mapping this and as minimum of the like gene 1 and gene 2 means minimum of this gene 1 and G2 will be the expression level of that particular reaction. And we did not resolve this OR relationship. So G1 or G2 means if either gene 1 or gene 2 cross the threshold, means that particular reaction will be considered as active. Then enzyme expression thresholds. So these 5 different thresholds were applied. Local T2 is the particular case, local T1 is the particular case of local T2, where we will not define this upper threshold, we will go on with the lower threshold alone. And these are this upper and lower threshold defined. And model extraction method, we there are several algorithms. We initially tried with these 2 algorithms, PASCOR and GIME, and after getting the context specific models, they were evaluated based on how these context specific models were able to recover these housekeeping reactions, and based on the cell consistency. I will define cell consistency later on slides. First, results going on how these context specific models, not context specific models, how these thresholding methods were able to recover the housekeeping reactions even though they have low expression. We have this 319 housekeeping genes. Took these 319 housekeeping genes from the Recon 2.2 model, and these were mapped to 929 reactions in the same Recon model. And so the thresholding methods were able to test the ability to recover these housekeeping reactions, how they define this active set, and whether the housekeeping genes are present in this active set or not. First, for the cancer cell line data, to define, these are those 44 cancer cell lines, and number of housekeeping genes recovered out of 929 housekeeping reactions in this Recon 2.2 model. So based on this plot, you can see that a global and local T1 doesn't perform well in recovering this housekeeping gene reactions. Gini, in terms of this cancer test, Gini performs better, marginally better than this stand-up method, which is recently published one. And these both, Gini and stand-up, are performing better than remaining all 3 threshold. Similarly, in the case of tissue data, stand-up performs better in recovering this tissue data, in recovering these housekeeping reactions. So what finally we can say is this: both the stand-up and local Guinea were able to recover this housekeeping reactions than comparatively to any other thresholding methods, even though those housekeeping reactions expressions values are low. So the second evaluation was based on the self-consistency. Before getting into self-consistency, let me explain what consistency is. Um, if we define this particular reaction to be active or to be a core reactions or context relevant reaction, what generally a model extraction method does is, or at least fast core does is, it's tried to make this reaction to be consistent. Means flux must freely flow through these reactions, which means if we, for reaction R2 to occur, we need D. For D to have, we need A and B. So whatever these reactions are required from the outside to taking substrate outside to get in outside, these reactions will be defined. If we say R2 to be active reaction, algorithm does by adding these reactions also in the final context specific model. So this is what consistency means. Most algorithms try to get this consistency method. Self-consistency means, what if my thresholding method already got this all reactions? Some thresholding methods might define only R1 and R2. Some might define all these 5. So if we are able to, through this thresholding method, if we are able to get all these reactions, consistent reactions, means there is no much work for this algorithms. So even my thresholding method does, or pre-processing step does most of the steps. So there is no workload to the algorithms which are there. These are the implementation details of fastcore and gimme. Gimme requires scores, and the scores were defined as given here, given a threshold. And local, and for self-consistency, we consider only 3 thresholding methods: local T2, stand-up, and local Guinea, as they perform well in this recovery of housekeeping reactions. And local T2 and stand-up were implemented as defined in their respective papers literature. This is self-consistency for fast core models. In fast core models, here the x is the fraction of reactions added, and y-axis is the number of cell lines. So in tissue data, again, local Guinea and stand-up perform better than this local T2. In terms of means stand-up performs marginally well than this. And in cancer cell lines data, local Guinea shows a better performance than other both thresholding methods. In the case of gimme models, stand-up doesn't work well in the gimme algorithm. Here almost more number of fractions were added by the algorithm than the what gimme is gonna add. Local Guinea and local T2 does well, and if we consider mean value, local Guinea does a better job than remaining both threshold methods. And to conclude my talk, thresholding is the most influenced parameter impacting the resulting model context and quality. Despite this, existing threshold methods have struggled to reflect the biological complexity. What these thresholding methods assume is that all genes have same patterns, regulations, and stabilities, but this is not the case. So local Guinea well captures this low expressed housekeeping genes, and models are more self-consistent. Models produced by this local Guinea are more self-consistent. And there is a drawback in this, not only local Guinea, on local thresholding methods. It requires huge data. As we defined, we need for more data to say that how much inequality does a gene possess, what is the Gini coefficient of this gene. If we have only single data, we have to rely on only the global thresholding method, and our future work is on implementing along with the constraint. As model extraction methods do require, it is our choice of providing constraints. If we have exomodulamics data, we can go ahead and provide the constraints to this exchange reactions, and implementing with the other discrete and continuous model extraction method. The discrete algorithms are what we tried now, gimme and fastcore. There are MBA and certain other algorithms also present for this discrete algorithms, for the discrete set of algorithms, where these generally filter out the reactions, irrelevant reactions. But there are another set of algorithms called continuous algorithms, where based on the omics data, we're going to play with this constraint on the flux, like example E flux and Atkinson's. They does this job, continuous job. So how we gonna implement this local Guinea on this both other discrete and continuous model extraction methods? Performance in gene essentiality predictions. We have this data for cancer gene essential genes. So we have to in future perform how this different thresholding methods gives says the particular gene to be essential and compare with the existing data. And all this housekeeping genes and gene essentiality, or even the cell consistence, were testing the thresholding methods based on the how whether they added a relevant reaction or not, context relevant reaction or not. But yet to no benchmarking method were actually developed also to test adding in addition of irrelevant reactions to the model, context specific model. So how can we whether say that a reaction is a false positive or it's a novel discovery? So that is one more aim of in our future. Thank you. Thank you, Nirav sir, for being there, and thank you, IBSE.
Thank you, Pavan, for the nice talk. There aren't any questions in the Q&A section. Does anyone in the panel want to ask a few questions? We have we have a question in the Q&A. Is the output of a local Guinea a set of reactions to be added in a context-specific model?
Yeah.
Okay.
Yes.
Okay. If there are no more questions, in the interest of time, we'll proceed on to the final talk of the workshop. So the final talk is by Rachita, who is a postdoc fellow again with IBSE. Her talk is on metabolic network analysis of microbial interactions, specifically on Klebsiella pneumoniae. And it is from the microbial modules on the International Space Station. So take it away, Rachita.
Hi, thank you, Shreyansh. I'll just share my screen. Is my screen visible?
Yeah.
Okay. So, hi everyone, good afternoon. I'm Rachita, a post-baccalaureate fellow at Dr. Karthik Raman's lab. And before I begin, I'd like to thank you all for being here. And I'd also like to thank IBSE for the opportunity to share our work with you today. So this is a project that I've been working on with Sanjay Balakrishnan and Amrita Mahesh at our lab, along with our collaborators at Caltech's Jet Propulsion Lab. So let me begin by sharing a couple of points on what we know on the International Space Station. So the International Space Station, or the ISS, is essentially a sealed closed system in outer space and has been around for nearly 20 years or so. So its environment has been classified as being extreme due to microgravity, high radiation levels, and higher carbon dioxide levels than what's found on Earth. And in spite of such extreme conditions, interestingly, there has been evidence of a large number of microorganisms that colonize its surfaces. And a recent study has even attributed this to routine arrival of crew members and payloads. So two recent publications in 2018 by Dr. Kasturi Venkateshwaran and Dr. Nitin Kumar Singh, our collaborators at JPL, and in 2019 by Chekinska et al., characterized these colonizing microorganisms in terms of their fungal and bacterial compositions. So they also further analyzed it for its spatial and temporal diversity across the station, and also identified some very important virulence factors and antimicrobial genes. So some of their findings were really interesting, and they now form the basis of our study. So to just give you an overview of how they conducted their study. So they sampled 8 distinct locations of the ISS at 3 different time points that were marked by the flights that brought these samples to Earth. So as you see here, flights 1, 2, and 3 essentially mean that the samples were brought from the ISS to Earth's surface. So yeah, okay, all right. So yes, so. Yeah, okay. So they were brought to Earth's surface. And the other thing is, as you see on the right, these are the 8 locations that were on the that were sampled on the ISS. And the locations from 1 to 4 are the ones that were frequented by the crew members. So here WHC refers to the waste and hygiene compartment, and ARED is essentially the exercise area. So just to get back to this, so what they essentially did is they sampled these 8 distinct locations across the 3 flights, and then they wanted to characterize the microbiome, like the viable microbiome from the dead cell. So they treated it with PMA, and then they did a shotgun metagenomic sequencing. So amongst these microorganisms, they found a high abundance of BSL2 pathogens. So this finding is pertinent because of a large number of studies that strongly suggest an increased antimicrobial resistance and virulence of these pathogens in outer space due to the effects of microgravity and space itself, as well as those that actually discuss compromised immune systems of the crew members that inhabit the space stations, again due to the effects of space. So as you can see in this table, which is actually from the paper that I previously showed you. So Klebsiella pneumoniae tops the list in 2 out of the 3 flights that they found. And as you know, that Klebsiella pneumoniae is one of the main causes of it causes pneumonia essentially, and it also causes a large number of other nosocomial infections, is widely studied for its multi-drug resistance, and is also studied for its hypervirulence that is characteristic of many of its strains. So this motivated us for our project, and we sought to gain insight into the interactions of Klebsiella pneumoniae in the distinctive microbial modules of the ISS. So metabolic interactions prevail in communities, and there are many microbial modeling paradigms as discussed in this monograph, such as graph-based we have, and then we also have constraint-based approaches, and there are also population-based approaches. So in our study, we primarily resorted to using a graph-based approach, and we employed the algorithm MetQuest, which of course, you may have also heard from Dinesh. So MetQuest is an algorithm that was developed in our lab by Dr. Arti Ravikrishnan, which is a novel dynamic programming algorithm to assemble the reactions into pathways from a given set of source molecules to a target molecule. And this can be used to answer or to understand how does one organism benefit from in the presence of another, so in terms of relieving the stuck reactions. So the figure on your right is a figure from Arti's paper. So on the top right, you see the individual metabolic networks of organisms 1 and 2. The orange metabolite nodes here are referred to the ones that are not present in organism 1, and similarly over here refer to those that are not in organism 2. And because of that, these reactions are crossed out in the sense that they are stuck because they are unable to proceed, as in they are, yeah. So whereas in the community metabolic network, we see that through a metabolic exchange over here, M10 from organism 2 to 1 and M9 from organism 1 to 2, we see that these reactions that were previously stuck have been relieved. So this extent of benefit that one organism receives from another can be quantified by the metabolic support index, which again characterized, which is essentially the fraction of reactions that are relieved in the presence of another. So the earlier papers that I had described had published the abundance of metagenomic sequence reads in all flights and locations. So we pruned this table to include only viable species that coexist with Klebsiella pneumoniae, and those that have a relative abundance of more than 1% at a particular site. So we essentially took those we took those whole genome sequences, and we used K-base to reconstruct draft genome scale metabolic models. Moving on, so we then calculated the metabolic support index values for every pair at a particular site that can potentially interact with each other. And just to give you, before I go into the microbial association networks that we constructed, just want to brief you on the legend that you can keep in mind for the upcoming slides. So over here, the node color and the size has been mapped to the percentage relative abundance of the organism in the site, and the edges have been mapped to the metabolic support indices. And so these are the sites where there was where Klebsiella pneumoniae was actually found to be present. So therefore, we considered these sites across these 3 flights for our study. So when we look at the association network in flight 1, we straight away can say that Panthea species, especially species like Panthea conspicua and Panthea agglomerans, seem to be highly dependent on some of the other organisms in the community, such as Escherichia coli and Klebsiella pneumoniae. We also observed that there is some slight dependence of Klebsiella pneumoniae on some of the fungal species, such as Penicillium rubens and Rhodothrula over here. And when we move on to the flight 2, we see that there is an increase in the number of Panthea species that colonize the location, as well as an absence of the colonizing fungal species. So actually the time difference between flight 1 and 2 is a time span of 2 months. And finally, when we move on to flight 3, which was sampled over a year later, actually, we see that there is a large number of Panthea species that colonize this location, and all of them seem to be highly dependent on the other bacteria such as Salmonella, Enterica, Escherichia coli, and as we earlier saw, Klebsiella pneumoniae as well. So just to give you an idea of what we observed, a sort of gist from all the other microbial association networks, in the interest of time, I will summarize it and say that some of the supportive species that we observed were the fungal species like Aspergillus rhodothrula, and also some of the Klebsiella, such as Klebsiella aerogenes and Klebsiella pneumoniae. Then we have Escherichia coli and Salmonella enterica. With the dependent species, we observed that almost all of the Panthea species were dependent with high MSI values. And as well as we also saw that Staphylococcus saprophyticus was quite dependent as well. So we then wanted to, we were then quite curious to actually see and understand what goes on in these metabolic exchanges. And in the graph here, the blue columns, as the legends show, represent a dependence, the number of organisms that are dependent on a particular metabolite. And the teal or the green column, the turquoise columns here represent the number of organisms that that provide a particular metabolite to support the community. So what we see is that many organisms are actually dependent on the metabolite D-glucanoate, which is actually provided by many of the fungi over here. And D-alanine, D-mannose, and as well as trehalose are are provided by many organisms in support to the community. So moving on looking at the metabolic requirements for each microorganism, there seems to be some kind of interesting trends. And of course, the same legend applies here as well, that the teal represents the metabolites that are given by a specific organism in support, whereas the blue represents the metabolite that is given, that a particular microorganism is dependent on. So on closely looking at the Klebsiella genus, what we observe is that unlike the other species, such as Klebsiella aerogenes, Klebsiella oxytoca, pneumoniae, etc., Klebsiella quasi-pneumoniae seems to only be dependent on many of the metabolites such as dealanine, deoxyguanosine, and urea, and actually does not provide any of the metabolites to support the community. And again, on looking at Panthea species, we observed earlier, if you recall, that Panthea are highly dependent in terms of their MSI values. We also see here that that they seem to take up many metabolites that other microorganisms can offer. And just to summarize and conclude, so we reconstructed draft genome scale models for species that coexist with Klebsiella pneumoniae. We then used a graph theoretical algorithm and calculated metabolic support indices of taking microorganisms pairwise in each site. And then we analyzed these metabolic exchanges and also identified certain metabolic dependencies. So currently what we're working on is strain-specific studies. We have the genomes of 10 Klebsiella strains that were isolated from the ISS. So we're trying to right now understand how they differ in terms of their metabolic capabilities, etc. We are also based on a suggestion that was given in one of the earlier reviews, we are also working on calculating the metabolic support index values in the presence of more than one organism. And finally, we are trying to identify whether reactions that are responsible for antimicrobial resistance are stuck, and if they're stuck, are they actually relieved in the presence of other organisms. So here are some of the challenges and limitations that we came across in when we were doing this analysis. So the first is actually, how do we best represent the environment of the ISS? As you know, there were 8 sites that were taken into consideration, and of course each site is going to have its own variations, but based on some kind of discussion, we identified and some suggestions from our collaborators. We have been using components of R2A agar and also minimal glucose media, and we are also continually renewing and modifying our environment so that it can best represent the environment. So the other thing is that what we have right now is draft reconstructions, and also the proper curation of these models will be quite difficult. And finally, that this is a static snapshot. There is actually a lot more that goes on, but we do believe that this will provide this will be a useful hypothesis for experimentation and will also form a good basis for further studies. So with that, I'd like to acknowledge my guide, Dr. Karthik Raman, my teammates, Sanjay Balakrishnan and Amrita Mahesh, and also all the members of the Computational Systems Biology Lab. I'd also like to thank our collaborators, Dr. Kasturi Venkateshwaran and Dr. Nitin Kumar Singh from Caltech JPL. I'd like to thank RBC DSAI for funding this project, and finally, I'd like to thank IBSE again for this opportunity. So thank you, and I'm now open to questions.
Thank you, Rachita. That was a nice talk. And do we have questions? We don't have questions in the Q&A. Do any of the panelists have any questions? If not, then I'll go ahead and ask you a question. So in the heat map where you had like dependent and supporters, I guess.
Yeah, right here.
Yeah, exactly. Yeah. So here, when you say dependent, do you mean that in the absence of like this metabolite, the organism won't survive? Is it an essential
No,
nutrient?
no, no. That is not necessary. That's a very good question, but no, that is. I mean, it's not necessary that in the absence of this metabolite, the organism will necessarily
Okay.
not grow. Yeah, we are still. I mean, we still have to filter these this list out and identify which are the most
Essential.
essential ones.
Okay.
Yeah,
Okay.
yeah.
And okay. Because like what I was thinking was a good thing to know would be even in this microbiome. So like I think even in Dinesh's case, in the ocular microbiome, there were many cases of something like this happening, but because you don't know if it affects if it is essential or not, you can't really make like the inference of presence of cheaters in the community. Like
Okay.
this particular block where you only have dark blue squares and no light blue. That means like they're just like feeding off of the community, so they're sort of cheating.
Hmm.
So yeah, so that might be something
Yeah.
useful.
Yeah.
Something
Definitely. Yeah. Yeah.
we can discuss this later.
Yeah.
So, oh yeah, we have a question. Oh, we don't have a question. So I guess that was it. Thanks, Rachita,
Thank
for the
you.
talk,
Thank
and we will move on to the closing comments. Uh, Philji, Himanshu sir.
Sorry, I heard my name. What's it? Shreyansh, I didn't get it.
Oh no, so Karthik sir messaged that he is on another call, so if any one of you can close the workshop.
Oh, okay, yeah. So I don't know money. He said money, but I can. So okay,
Sure, yeah.
yeah. So great. Thanks everybody. I think these are very interesting talks. It shows the breadth and the diversity of the research work that goes on at IBSE, and thanks to all these young researchers who put their hearts in sort of solving some of the problems that either they come up with themselves or sort of come up with when they discuss it with the PIs. So I'm hoping everybody who attended today's workshop did get the feel of what goes on at IBSC and how we are trying to utilize both real world problems as well as some synthetic problems like simulations and all. And I thank again everybody and for keeping the time for sure, and Shreyansh for making sure. I was seeing that he was like on the clock for many of the speakers. He was saying you're over time, so thanks Shreyansh for doing the timekeeping and keeping the clock. And thanks to Phil G. Philip, who actually helped organize this workshop from the beginning, and I think without his support, it would be very difficult to run many of the activities that we carry out at IBSC. So thanks very much, and you all stay safe and have a nice weekend.
Thank you, Himanshu.
Thank you, sir.
So, Phil G, you can close the live streaming for sure.
Yeah, sure.
Thanks, guys.